The overall goal of this high-throughput assay is to provide a simple, efficient, and scalable platform to identify genes and neurons that control a simple choice behavior in Drosophila.
The main advantage of this method is that it can efficiently assay egg choice and single animal resolution and in high throughput manner.
To begin the experiment, place 8 females and 6 males into a narrow food vial filled with standard fly media. Then use the female progenies grown from this vial for the egg-laying experiments. 2 to 3 days after female eclosion, make fresh yeast paste by mixing 6 grams of active yeast with 10 milliliters of 0.5% propionic acid. Use a spatula to apply the yeast paste onto the side wall of the food vials. Next, place 30 previously collected eclosed females and 20 males into the vials. After 4 days, check the surface of the food medium. If the surface is wet from the larvae actively burrowing in the food, then the female flies are ready for egg-laying experiments. Continue with the experiment by obtaining previously constructed acrylic egg-laying chambers. Insert plastic sheets into the top, loading a piece of the chamber to serve as the bottom surface. Next, anesthetize the females on a carbon dioxide pad and load them individually into each egg-laying arena. Allow the flies 30 minutes to recover from the carbon dioxide and to acclimate to the new environment. While the flies are adjusting, fill a tube with agarose up to the